Millions of years of evolution have endowed our bodies with a natural ability to fight disease. Our immune systems have a remarkable and ferocious mechanism to identify foreign invaders such as bacteria and viruses and destroy them. For over a century, scientists have wondered whether this natural mechanism could be harnessed to combat disease. Could a magic bullet be made that could selectively identify and destroy the disease agent itself? At the heart of this process is the antibody. 40 years ago, Nobel Prize winner Cesar Milstein and his team at Cambridge Laboratory for Molecular Biology built upon this basic idea to develop a technique to produce bespoke monoclonal antibodies. Antibodies that are produced by identical immune cells from a unique parent cell. Once monoclonal antibodies have bound to their target, they mark them for destruction by the body's natural defense mechanisms. This pioneering research was the inspiration for the development of a remarkable new drug that had the potential to treat a whole range of diseases, from leukemia to arthritis, cancer to multiple sclerosis. This is the story of Campath 1H. In the late 1970s, Professor Hermann Waldman, then a lecturer in the Department of Pathology at the University of Cambridge, and now head of the Sir William Dunn School of Pathology in Oxford, was applying for his first Medical Research Council programme grant.
Well, I came into immunology, and in fact I came to Cambridge because I wanted to do research as a medic. I am a medic, because I felt that immunology offered some treatments, possible treatments for diseases that had unmet needs. Immunological diseases represent about a third of the most important diseases in the Western world. And we don't have good treatments. We have steroids, we have all sorts of drugs that just penalize the whole of our immune system and damage our organs, our kidneys, to try to stop immune attack. And the fact the body can do it itself told a lot of us that if we could learn how to harness the way the body does it and exploit the way the body does it, then we are going to be much better off than using these nasty drugs.
The immune system is a network of many cell types that protect the body from bacteria and other disease-causing organisms called pathogens. When a pathogen enters the body and infects human cells, immune system cells that circulate in the blood, called T and B lymphocytes, detect its presence by binding to antigen chemicals on the surface of the pathogen and become activated. The activated T lymphocytes kill the pathogen-infected cells directly, or help the activated B lymphocytes to make antibodies, secreted proteins that recognize specific antigens. These antibodies coat the pathogens, which labels them for destruction by other immune system cells. It does, however, have an Achilles heel: the loss of immunological tolerance.
Immunological tolerance normally refers to the situation by which the immune system decides that it's not going to react to something that it should not, ourselves for example, but it should react to a foe, a potentially dangerous foe like a microbe, a virus or a bacterium. It's very important that it has that discriminatory ability, because in no way should it attack self. Many of the diseases we are trying to tackle are actually where the immune system has failed in its ability to keep tolerant of self and by mistake attack self. It's the price we pay for having a vigilant immune system. Immunologists have been trying to investigate immunological tolerance for a very long time. It's a really challenging problem, because once a person is delivered from the womb, their immune system has already become tolerant to a lot of the self-antigens. So studying tolerance is actually quite hard. It's much more difficult than studying immunity, because it's a fait accompli at birth, whereas immunity, you just come along with something foreign, inject it, and then follow the response from the time. So the field has depended on all sorts of quite ingenious approaches to study what goes on. Because our goal was therapeutic, our approach was to say, can we produce immunological tolerance to something that would normally be a foe, and now make it appear as a friend?
The approach Waldman took was to make an anti-lymphocyte antibody using a technique that had recently been developed by César Milstein at the MRC Laboratory of Molecular Biology in Cambridge. By fusing myeloma cells, cancer cells that develop from B lymphocytes, with cells from the spleen, Milstein had managed to make cell lines that indefinitely produced large amounts of a single antibody, known as a monoclonal antibody. Such monoclonal antibodies were ideal for Waldman's experiments and were given the code name Campath, standing for Cambridge Pathology. An immediate medical application became very clear: to improve bone marrow transplant in patients whose blood systems have been destroyed by leukemia and radiotherapy.
So the idea was, can we get an antibody that would kill the bad cells, leaving the stem cells that give a new blood system free to work? That was the basis of it. And we were very fortunate in that we found Campath was an antibody that would spare the stem cells, but hit the bad cells and allow the marrow transplants to occur. So we knew early on in the 80s we had something that was going to be a good agent to hit the lymphocytes. And the argument was, if we could find a way to knock someone's lymphocytes down transiently to a very small number, we might interrupt the disease, and then when that number of lymphocytes is very small, they may have a chance to be reprogrammed so that they no longer look at what they're reacting to as a foe. And we had all sorts of theoretical arguments and experimental evidence that suggested that an immune system without many lymphocytes might be more prone to tolerance.
Late in 1979, Waldman and his team, including Drs. Mike Clark and Jeff Hale, immunized a rat with human lymphocytes and fused its spleen cells with a rat myeloma cell line. Over Christmas, Waldman isolated several antibody-producing cell lines from this Campath-1 fusion, and his team set to work to identify an antibody that could kill mature T lymphocytes without damaging the bone marrow stem cells. The monoclonal antibody that best met these criteria was an immunoglobulin M antibody, an antibody that is produced by B cells. B lymphocytes can make several different types of antibody, each of which behaves differently in terms of which immune effector mechanism it interacts with to destroy pathogens. The first bone marrow transplants that used Campath-1M for T lymphocyte depletion were performed in the early 1980s.
We were, first of all, looking for an antibody that would help people who have bone marrow transplants. Special problem in bone marrow transplants called graft versus host disease. We developed some antibodies that would remove the lymphocytes from the bone marrow and prevent that disease.
And what all we cared about was that the person should be able to get a blood system growing out of the bone marrow transplant without the biological warfare.
But these antibodies didn't work in patients for treating leukemia or diseases that we wanted to treat in patients.
Waldman and his team returned to the lab to find a better way to suppress the patient's immune response.
What we had to do was to find a form of that antibody, Campath-1, that could work inside the patient. So it was one thing to be able to take out the lymphocytes in the test tube, but we had to find a form of the antibody that could work in the patient to take the lymphocytes out in the patient. And we found actually that we needed a different variety, a different form of that antibody to do the same.
And we discovered that we needed to change the antibody from one type, a type that we called IgG2A, into a different type, a type that's called IgG2B. And I spent a lot of time culturing cells that made these antibodies, and we grew many of the cells. We selected many different clones, and in the end I screened around about 100 million clones, and we found one that had switched just spontaneously from making the 2A sort to the 2B sort, the sort that we really wanted.
Campath-1G, like the original Campath-1M and Campath-1H, binds to a molecule called CD52 that is present on lymphocytes and some other human cells.
The very first patient the 2B antibody was tested in was a patient with leukemia. And the antibody, we manufactured the antibody in our laboratories in Cambridge. We made enough of it to treat the patient, and the patient was treated at Adam Brooks Hospital, And it was the first time that we saw leukemia cells being cleared from the blood and really disappearing not only from the blood but from the bone marrow of this patient. It's a very exciting time for us. And I remember Hermann coming into the lab and saying, Jeff, it's really working. It's almost like a miracle. It's worked so well, better than we thought. And we were very excited about that. And one of the big problems was that the antibody had originally been made in rats. It was a rat protein. And so if people were treated with it for some time, they started to make antibodies of their own, anti-antibodies, if you like.
In the case of Campath, given short term, maybe that isn't a big problem. But as a generic issue, if you wanted to give repeat treatments, you would like to make the antibody as human as possible so the patient would not reject something that looks a lot like their own antibodies.
Clearly, a more human-like antibody was needed that would be ignored by the immune system. Fortuitously, another Cambridge scientist, Professor Sir Greg Winter, had just developed a way to humanize antibodies.
We had the idea that maybe we could take a mouse antibody and just transplant those little bits from the mouse antibody that are responsible for binding to the target and put those into a human antibody. And in that way, transfer the binding activity of the mouse antibody into a human antibody. And that's what became known as humanization of antibodies. So we started off on a series of what we call model antibodies, antibodies that weren't any clinical use. And these worked, our technology worked. And we thought, therefore, maybe it was worth looking for a therapeutic antibody that we could humanize. And Cesar Milstein, who was the head of division and in fact who'd invented monoclonal antibodies, suggested to me that I should talk to Hermann Waldman, who was working across in the Department of Pathology at the Addenbrooke site in Cambridge. So I went and had a talk to Hermann, and we set up a very nice collaboration where we largely did the genetic engineering and making the antibodies, and he was involved in testing their properties. In fact, it was Lutz Riechmann from my group and Mike Clark from his group were the people who were largely doing the work. And they did a great job, and they showed that these humanized antibodies could be as potent at killing cells as the original rat antibody that he'd started with.
The first lady to be treated with Campath was a lady suffering from leukemia. And this time, when we treated her with the Campath, the humanized form of Campath, unlike the rat antibody, she made a really good response, which lasted for a very long time. And after some months, lymphocytes started to come back in her blood, but for the first time, these were normal, healthy lymphocytes rather than tumor cells.
When we were able to get this antibody into patients, and in fact, again, this was largely worked by Hermann with his colleagues at the hospital, Martin Dyer and Robert Marcus, who put this antibody into patients with non-Hodgkin's lymphoma. And it had an absolutely dramatic effect, blowing away several kilograms of spleen. And that was absolutely dramatic to me, because I hadn't previously thought that the kind of work I was doing could lead to that kind of patient benefit.
The clinical grade material needed for these studies was produced in the Therapeutic Antibody Centre that Waldman set up in Cambridge in 1990.
So in 1990, we had a great opportunity to start working with the Blood Transfusion Centre on the Addenbrooke site, and they offered us some space to build a manufacturing facility. So that we called the Therapeutic Antibody Centre. Our plan was to make a whole range of antibodies for treating different sorts of diseases, and we were starting with Campath.
The development of a drug for clinical use is a highly regulated, long and expensive process, so drugs only get to market if pharmaceutical companies become involved in their development. In the early 1980s, the Cambridge researchers assigned the rights for the Campath-1 cell lines to BTG, originally a government body set up to facilitate the exploitation of inventions from UK academics, but now an international specialty healthcare company. When the basic research being undertaken by Waldman's team suggested that Campath-1G and Campath-1H were more likely to have a clinical future than Campath-1M, these antibodies were also licensed to Wellcome Biotech. Indeed, once Campath-1H had been handed over, the company abandoned its work on the earlier antibodies and started a program of clinical trials of Campath 1H in rheumatoid arthritis, leukemia, and lymphoma. Meanwhile, the academic scientists continued with their basic research, refining and extending their understanding of how Campath 1H was working in various diseases by collaborating closely with the physicians who were giving the antibody to the patients. This research was helped along by the development of new molecular techniques and by improved understanding of the human immune system.
After a period of years, Hermann moved away from Cambridge, and he had an offer of a job in Oxford. And he asked me if I'd like to join him. And I said, well, it would be nice to come to Oxford if I can have a bigger and a better antibody centre, because we were starting to outgrow the centre that we'd built at the Transfusion Centre here in Cambridge. And so we did move to Oxford in about 1995, and we did have a bigger centre there, and it was very nice. But just again, at the opening of that second centre, we had a second blow, and that was that the commercial production of Campath was going to be stopped altogether, and Wellcome decided to abandon the work because it hadn't been so successful for treating arthritis as they had hoped, and they hadn't been doing the trials in multiple sclerosis. They hadn't seen the good results that Cambridge has seen in MS, and they decided to abandon the project. So that was a huge disappointment.
Although the antibody worked well in some types of leukemia, it did not work well in all leukemias, and in the rheumatoid arthritis trials, Campath 1H had permanently suppressed patients' immune systems. This long-term effect was in part the consequence of treating patients who had already been treated and failed on other drugs. This decision was very disappointing for Waldman and his colleagues, who strongly believed in the clinical potential of Campath 1H. Undeterred, Waldman took an objective view and responded by looking at the evidence, figuring out what had gone wrong and finding ways to put it right rather than giving up. In 2001, Campath 1H was approved in the US for the treatment of B-cell chronic lymphocytic leukemia in patients who had failed to respond to conventional chemotherapy. The license for Campath 1H traded hands several times until finally, in 2004, Genzyme Corporation acquired the production rights.
Time went by. We went on working with the Adam Brooks team, particularly with Alistair Compston and his team working on multiple sclerosis, and with Professor Roy Cowan and his team who were doing kidney transplants, and they were using Campath for preventing rejection of kidney transplants. So there are a number of different clinical trials in different places around the world, but particularly here in Cambridge.
During the late 1980s, it was becoming clear to researchers that MS is an autoimmune disease, a disease in which a person's immune system attacks their own tissues.
So multiple sclerosis is the commonest potentially disabling disease of the brain and spinal cord affecting young adults. The illness targets the myelin sheath, which surrounds nerves in the brain and the spinal cord, eventually damaging both. For people who have multiple sclerosis, the illness usually starts as attacks which come and go, and they're well in between, but later those attacks reduce in number and they're replaced by slow progression, so that in the end the affected person faces both physical and mental disabilities. Multiple sclerosis is characterized as an immune disease for two main reasons. First of all, because if we look at affected tissue, we see evidence for chronic inflammation of the brain and the spinal cord. And secondly, animal models of multiple sclerosis have shown that transferring components of the immune system will indeed reproduce the disease.
We began to wonder whether we could help patients with multiple sclerosis by preventing the movement of activated lymphocytes from their bloodstream into the brain and spinal cord.
Treatment with Campath 1H looked like one way that this could be achieved, and in 1991, Compston started an 18-year-long collaboration with Waldman and his team by trying this approach for the first time in a woman who had developed MS some years earlier.
When we first started treating people with multiple sclerosis, there weren't any treatments for the disease, and our initial efforts were also unsuccessful. It was clear that the drug did what was being asked of it. It did remove immune cells from the circulation.
Keeping in close contact with Waldman's team, Comston and colleagues carefully followed the progress of their patient for about 18 months before treating another 6 people.
By 1994, we had satisfied ourselves that Campath 1H treatment would stop the development of new inflammatory lesions in the brain, and in addition, it seemed as though our patients were having fewer new clinical attacks after the treatment.
By 1999, Comston and a clinical trainee, Dr. Alistair Coles, had treated 27 patients, all of whom had already entered the secondary progressive stage of MS.
We paused to analyse our results, and we realised that although we had stopped the inflammation in the brains of our patients, and that had led to them having no further attacks of the disease, unfortunately their disability continued to progress. That is, the problems that they had prior to receiving Campath just gradually got worse.
This observation puzzled the researchers. If MS is an inflammatory autoimmune disease, why was Campath 1H treatment failing to help people in the progressive phase of the disease, even though the treatment seemed to turn off inflammation?
What we realised is that disability in people with progressive multiple sclerosis continues because nerve cells wear out, they degenerate. And Campath hadn't helped them because it isn't inflammation then and there that's causing this degeneration. We speculated that this degeneration was due to damage done to the nerve cells some years before, during the relapsing remitting phase of the disease, when inflammation is the predominant process. So we concluded from this that for Campath to be effective, we really had to treat people much earlier on in the course of the disease, when inflammation was the main problem, to prevent nerve damage, which ultimately might lead to nerve degeneration.
Comston and Coles now began to treat some of their patients suffering from relapsing remitting MS with Campath 1H.
We saw the same thing in that inflammation in the brains of these people was suppressed, and as a result they had no further attacks. But what was different now is that their disability not only did not get worse, but it actually improved. So our hypothesis turned out to be correct.
It was time to take Campath 1H into formal clinical trials to prove the drug's efficacy and prepare the way for marketing the drug for the treatment of MS.
So to do a formal clinical trial in people with relapsing remitting multiple sclerosis requires a lot of funding. So we needed a commercial partner, and with Ilex Oncology and then with Genzyme we found companies who were supportive of our thinking. And so a phase 2 trial was set up and eventually reported in 2008.
Patients enrolled in the trial had to meet strict entry criteria and were treated with up to 3 annual doses of Campath 1H. The effects of the drug were measured 3 years after the initial treatment.
And here we saw confirmation of our findings. Treating people early on in relapsing remitting MS results in reduction in their attacks and not only stabilisation but actually improvement in their disability. The next stage of testing a drug is a phase 3 trial, and we compared Campath with active therapy, a standard drug, beta interferon, in 2 separate phase 3 trials. The difference between them being that in one trial we looked at people with multiple sclerosis who had never had any other previous drug, and in the other trial we looked at people who had already been on a standard therapy and had had some breakthrough of their disease. And in both trials we saw that Campath reduced attacks of multiple sclerosis more effectively than the standard treatment beta interferon. And in the second of those trials we also saw that Campath reduced the risk of becoming disabled and indeed improved disability in people who had already been on a standard therapy and had had an attack of their disease. And it's this result that is unique. There is no other drug in the treatment of multiple sclerosis that has shown efficacy over and above the standard first-line treatments in this treatment-resistant population.
As effective as Campath is at treating multiple sclerosis, like other drugs, it does have side effects. In particular, the risk of developing another autoimmune disease. 30% of our patients develop either an over or an underactive thyroid gland, and less than 1% develop a disease in which the platelets of the body's clotting system are destroyed, leading to an increased tendency to bleed. The good news is that these side effects are easily treated, and because of our research, we know what to look out for, and we may be able to predict, or even in the future, prevent them.
So where we see Campath is in treating people really early on in the course of their illness, when they don't necessarily have any obvious disabilities to see, but they're beginning to lose their fine motor skills or their fine intellectual skills. And we hope to restore them to exactly where they were before. And a good example of this is the case of Tony Johnson, who, when he developed MS, you wouldn't have known it other than the fact that his golf couldn't be as excellent as it was before.
Yeah, I woke up one day in 2003, my left hand was numb. Thought I'd been lying on it awkwardly, and then as the day went on, my whole left side went dead. And I went and saw a specialist the following day, and I was told I had a viral infection. I was given steroids, and then things just got worse and worse, and 6 months later I was diagnosed with MS. So, once you get told that you've got MS, it's one of those things that happens to other people, and nobody really understands or knows a lot about MS until you get it. I got very lucky. I saw a specialist in London by the name of Gavin Giovanni, and he suggested that I get on the Campath trial at Addenbrooke's Hospital, which was, turned out to be the most fantastic break, because I was told I'd never play golf again. And, I had all these ghosts in my mind about being invalided and, ending up in a wheelchair. And thankfully, the Campath has put paid to that. I'm out competing again on the European Seniors Tour. I just got very, very lucky.
If all goes well, Genzyme expects to receive UK approval for the use of the drug to treat MS in 2013, 21 years after the first patient with MS was treated with Campath 1H, and 33 years after Waldman's team started the research that produced Campath 1H.
Well, looking back over the last 20 years during which we have been developing this program, it is, I think, a reasonably good example of what I like to call clinical science, or what is now called experimental medicine, in which the approach is to treat patients, to see what happens, and to learn from that experience in order better to understand the illness. We did face struggles along the way. It was difficult to sell the hypothesis and to explain the risks of what is a radical treatment of the immune system professionally, to funding agencies, and to the pharmaceutical industry, who owned the rights to the drug at different times. And perhaps if those very understandable hesitations had not been there, we might have been where we are now, say 5 years ago. But in the end, the results speak for themselves, and it's for physicians and neurologists to decide how best this drug is used in a market where there are already a number of other treatments for multiple sclerosis.
We always wanted to be able to use the body's killing mechanisms, damaging mechanisms, and the antibody to be able to recruit them to kill what we wanted. And we were pleasantly surprised that that was possible. The second thing that was great was being able to actually go to the clinic, because that had always been my intention. And being able to demonstrate that, in fact, we could deal with some of these unmet medical needs, albeit slowly, and you had to for ethical reasons, but gradually we could see that we were going in the right direction, offering patients something that wasn't available before.
A couple of years earlier, I'd been a basic scientist, and then suddenly find myself with this molecule that we'd constructed together being put into patients, and it was terrific. And in fact, that was not only it was transformative in the impact it had on the technology, but also transformative for the kind of my later career development, which was to very much focus on trying to look at other approaches to making therapeutics.
I have been working on Campath as a treatment for multiple sclerosis since 1994, and it has been a huge adventure.
Looking back over the work with Campath 1H, it's been very enjoyable and a privilege to work with so many distinguished scientists and clinicians who've enabled the work. But it's been the contact with affected individuals, the patients, who've put their hopes and fears on the line, which has been so very inspirational.
The journey from fundamental research on the immune system to a commercial product has been a long one. It's a testament to the commitment and determination of the researchers that 40 years after the discovery of monoclonal antibodies in Cambridge and 30 years after the early studies on Campath, we are on the brink of a revolutionary new treatment for MS.